Okay, let's continue. Ribosomes, the protein factories. Ribosomes are responsible for protein synthesis. They translate genetic information from the messenger RNA, mRNA, into chains of amino acids, which fold into functional proteins. Many of the enzymes required for both photosynthesis in the chloroplast and cellular respiration in the mitochondria are produced by ribosomes. Therefore, the ribosome supports energy flow by building the very machinery that makes and uses energy. In summary, chloroplast converts solar energy into chemical energy, which is the glucose, which is then converted by mitochondria into a usable form, ATP, for the cell. Ribosomes are essential in this cycle because they synthesize the proteins and enzymes necessary for both of these critical energy producing processes. Number 3. Design a diagram showing the connection between the nucleus, DNA, and protein synthesis in cells. So this is the diagram. You see there the nucleus, and you have there the ribosomes, and then translation, and then you have um, the chains of the mRNA, the tRNA, and then the transcription, and then there's the protein. Protein synthesis is a two-step process that starts with the genetic information stored in the DNA within the nucleus and ends with the creation of proteins by ribosomes in the cytoplasm. The role of the nucleus and DNA. The nucleus serves as the cell's control center, housing the DNA. The DNA contains the blueprint for all proteins a cell needs to function. The first step of protein synthesis, transcription, takes place here. During transcription, a specific section of DNA, a gene, is copied into a mobile molecule called messenger RNA. This is crucial because DNA itself is too large to leave the nucleus. The role of ribosomes. The messenger RNA molecule then travels out of the nucleus and into the cytoplasm, where it finds a ribosomes. Ribosomes are cellular machines that perform the second step of protein synthesis, called translation. The ribosomes reads the genetic code on the messenger RNA, and with the help of the transfer RNA, tRNA molecules, assembles a chain of amino acids in the correct order to form a polypeptide chain. This chain then folds into a functional protein. Practice problem number 4. Show the structural relationship between enzymes and substrates and explain how this determines their biological function. So we have here a substrate and then an enzyme with its active site. So using the lock and key, then you have there the enzyme substrate, the complex one. And then the other one using induced fit. So you have the um, complex enzyme substrate. The structural relationship between an enzyme and its substrate is based on the concept of a highly specific fit, which is essential for the enzyme's biological function. Enzymes are biological catalysts, and their unique three-dimensional structure allows them to bind to a specific substrate molecule, forming a enzyme's substrate complex. This relationship is explained by two primary models. The first model is the lock and key model, and then the other model is the uh, lock and fit model. So this model proposes that the active site of an enzyme is a rigid structure that perfectly matches the shape of a specific substrate, just as a unique key fits into a specific lock. The substrate fits precisely into the active site. This model explains the high specificity of enzymes, but it doesn't account for the flexibility of proteins. The induced fit model, a more widely accepted model. The induced fit model suggests that the enzyme's active site is not rigid. Instead, it is a flexible structure that slightly changes its shapes to fit the substrate more precisely once the substrate binds. This conformational change helps to cradle the substrate and put stress on its chemical bonds, lowering the activation energy required for the reaction to occur. This dynamic interaction explains how the enzyme not only binds to substrate but also actively facilitates with the chemical reaction. How structure determines function. The unique structure of an enzyme is crucial for its function in the following ways. Letter A. Specificity. The specific shape of the active site determined by the folding of the enzyme's amino acid chain allows it to bind with only one or a few specific substrates. This ensures that the correct chemical reactions occur at the right time in the cell. Letter B. Catalysis. The precise arrangement of amino acids in the active sites lowers the activation energy of a reaction. This can be viewed by, this can be achieved by orienting substrate correctly so they can react with each other, straining the bonds within the substrate and making them easier to break. creating a favorable microenvironment. For example, acidic or basic for the reaction. Once the reaction is complete, the products are released and the enzyme returns to its original state, ready to bind to another substrate molecule and catalyze the same reaction again. Practice problem number 5. Create a diagram of the respiratory system, labeling structures, and indicating how each part contributes to gas exchange. So there you are, the respiratory system. We have there the nasal cavity plus and paranasal structures. We also have the nostrils, the larynx, the trachea, the right main bronchus, the right lung, the diaphragm, and then the left lung, and then the bronchi, the left main, and then the oral cavity, and then the pharynx. The human respiratory system is a network of organs and tissues that work together to perform gas exchange, which is the vital process of taking in oxygen and expelling carbon dioxide. This process occurs as air travels through a series of structures. Structures and their role in gas exchange, the nose and mouth. These are the entry points for air. Hairs in the nose filter out large particles, and passages warm and humidify the air to prevent damage to the delicate lung tissues. This prepares the air for its journey to the lungs. Trachea, the windpipe. The trachea is a rigid tube supported by rings of cartilage that prevent it from collapsing. It acts as a primary passageway, transporting air from the pharynx to the lungs. Bronchi and bronchioles. The trachea branches into two main bronchi, which enter the lungs. These then branch repeatedly into smaller and smaller tubes called bronchioles, forming a tree-like structure. These airways continue to warm, moisten, and clean the air while distributing it to the gas exchange surfaces. Alveoli. At the end of the bronchioles are the millions of tiny balloon-like air sacs called alveoli. This is the primary site of gas exchange. Each alveolus is surrounded by a network of tiny blood vessels called capillaries. The walls of the alveoli and capillaries are only one cell thick, allowing for rapid and efficient diffusion of gases. We will have the next part of our quarter 3 lesson biology lesson 2.